let us use this lockdown period as a recap session for our immunology, and this is for the second year students, and I would like to start with vaccines. So the vaccines are to provide immunity. Immunity, as all of you know, is a state of protection against the microorganisms. So there are two ways to get the immunity. One is an active immunity, the other is a passive immunity. In active immunity, you can see, as the name implies, our immune system is actively involved. Whereas in passive immunity, it is the direct administration of antibodies without the involvement of immune system. And in this active immunity, our B cells and T cells react with the antigen and produce antibodies and cell-mediated immunity. Whereas in passive immunity, antibodies raised in horse or in other species will be administered directly to us in order to get a state of immunity. Now, the another important difference between the active immunity and passive immunity is active immunity offers long-term protection as your cells are directly involved. Whereas in passive immunity, it offers short-term protection. And vaccination comes under active immunity as it triggers our immune system to produce the antibodies or the cell-mediated immunity to give us a state of protection. Right. So the next thing is about the history of a vaccination. The discipline of immunology itself has started from the history of a vaccination. Whenever we thought about the history of vaccination, we would come across two prominent names. One is Edward Jenner, the other is Louis Pasteur. So first let us see the contributions of Edward Jenner, then we will move on to Louis Pasteur. These are two simple experiments carried out by the two famous scientists which led to the discovery of vaccination. So first let us see the contributions of Edward Jenner. So Edward Jenner observed that the milkmaids who were getting the cowpox are not getting the smallpox. Smallpox is actually a severe form of disease, whereas a cowpox is the milder form. Milkmaids who are working with the cows are getting a disease cowpox, but they are not getting a much more dreadful disease, smallpox. So Jenner observed that getting cowpox is making them immune from getting the smallpox. This is the reason why he has applied the pustules of cowpox directly into the individuals in order to get the immunity from smallpox. So into those individuals whom he has ingested the pustules are challenged with smallpox virus. Then surprisingly the individuals are getting immunity to smallpox. So he reasoned that the pustules from cowpox are providing protection to the individuals from not getting the smallpox. Now, let us see the contributions of Louis Pasteur about almost 100 years later. Louis Pasteur was working with fowl cholera and he isolated the bacterial culture responsible for the fowl cholera and injected into the fresh batch of fowls. All the fowls are getting the disease. Then what he did is, after coming from the vacation, the same old bacterial culture was used to infect the fresh batch of fowls, and these fresh batch of fowls are not getting the disease, and even if they have got, they have recovered from it. Now the last part of the experiment is, into the same fowls which were recovered, he injected a much more fresh culture of bacteria which is more virulent, and surprisingly even now the fowls are not developing the cholera but are immune to them. So then Pasteur has postulated that the weakened culture of the fowl cholera bacterium is somehow protecting the fowl from getting the immunity when the fresh culture of bacteria is ingested, which is now became the principle of vaccination. The basic principle of vaccination is, whatever be the microorganism we want to get the immunity against, we have to weaken the microorganism first, which is responsible for the disease. I will tell you how to weaken the microorganism, but first let us concentrate on the principle. We have to weaken the microorganism responsible for the disease so that it has lost all its pathogenic ability, but its structure is intact. so that it can produce an immune response into the individual when you inject it into him. So that now when a real microorganism attacks him, he will be ready with the protection in the form of either antibodies or the cell-mediated immunity, or in most cases both. Right? So that is the basic principle of vaccination. Now the next point is how to weaken the microorganism, and depending on this we have two different types of vaccines available. One is attenuated vaccine, the other is inactivated vaccines. Now let us see different types of vaccines. First and foremost category in them is whole organism vaccines, where the whole organism responsible for the disease is used. And then the next category is purified macromolecules, where it is not the whole organism but some parts of it, like some epitopes of it is used. Next, the third are DNA vaccines, fourth are recombinant vector vaccines, and the fifth are multivalent subunit vaccines. These are specialized vaccines which are in use for some special purposes. First, let us concentrate on the whole organism vaccines, which are again of two types. One is either attenuated and live, the other is inactivated or killed. So we will see the whole organism vaccines. The first category in vaccines are whole organism vaccines, where the whole organism responsible for the disease is used. And we will take the whole organism and then either we will kill it by heat or chemicals, or we will weaken the microorganism so that it has lost its pathogenicity but the structure is intact. So now first let us see how to prepare an attenuated vaccine. Attenuation is about to produce the microorganism with its weaker version. So first, how to produce a microorganism with a weaker version or with a reduced pathogenicity. So we can do that by culturing it in abnormal culture conditions, so that whenever it is injected into a normal individual, it cannot produce a disease but it can grow. So that is the process of attenuation. On the other hand, we have another vaccine, another type in the whole organism vaccine, that is inactivated vaccines. Inactivated vaccines are prepared by killing the microorganisms either by heat or by chemicals like formaldehyde. Here you do not have any microorganism that is alive. We are going to kill the microorganism. That's why the vaccines are named as inactivated vaccines. Attenuated vaccines contain the live microorganisms with reduced pathogenicity but with structure intact. We can produce attenuated vaccines by culturing the microorganism in abnormal culture conditions. Right? I will explain it later, but do remember we are going to prepare attenuated vaccines by culturing the microorganism in abnormal culture conditions so that it cannot grow in normal conditions. Now, but here there is a difference between attenuated and inactivated vaccines. What is the vaccine that we have to choose? So depending on the purpose, we can use either attenuated version or an inactivated version. Both has their advantages as well as disadvantages. So first let us see the advantages of attenuated vaccines. So here the advantage of attenuated vaccines is it can produce both humoral and cell-mediated immunity. So it can trigger your T cells as well as B cells. So the immune response produced from attenuated vaccines will be quite effective. And another advantage is while using the attenuated vaccine, there is no need of multiple boosters. That means you need not administer the vaccine again and again. You have to administer it once and then you have to supplement it with one more dose. That is enough because it will trigger both the branches of immune system and the protection you are getting is going to be long lasting. Whereas when coming to the inactivated vaccines, they will produce mostly the humoral immunity and also they do require multiple boosters. As because they cannot trigger your immune system properly, you need to administer the inactivated vaccines more than once. In fact, they need multiple boosters. These are the advantages associated with attenuated vaccines. Now let us see the disadvantages associated with attenuated vaccines. So these particular attenuated vaccines are going to be unstable when compared with inactivated vaccines. And another most important thing is, as the attenuated vaccines contain the live microorganism with reduced pathogenicity, of course, but still there is a chance that they can revert back to their virulent form. That means they may cause a disease rather than relieving you from the disease. They may cause a disease, so that is the danger associated with the attenuated vaccine. Whereas in inactivated vaccines, there is no such hazard. They are stable, and even they cannot revert back to virulent form because you have already killed the microorganism. So here, depending on your purpose and depending on the microorganism associated, depending on the disease associated, you can choose either attenuated or either attenuated or inactivated vaccine. But remember, whatever be the mechanism, they do contain whole organism. That is the complete microorganism. Either you are going to weak or you are going to kill. Depending on that, we have two categories. But because of these hazards associated, we are going with the another vaccine, that is about purified macromolecules used as vaccines. That means here there is no question of any revert back to virulent form. We are not using the whole microorganism because of the potential danger associated with it. So what we are going to do is, whatever be the epitopes that are responsible for causing disease, we are going to isolate it, and then we are going to weaken them, and then we are going to produce a vaccine against it, and they are called as purified macromolecules as vaccines. That is the second category. We will see them now. Let us see how to prepare an attenuated vaccine which consists of a microorganism but with reduced pathogenicity and all its structural epitopes intact. So let us talk about a vaccine for TB, that is tuberculosis, and the causative organism is Mycobacterium bovis. So I have to prepare a vaccine for TB which is caused by Mycobacterium bovis. So now what I will do is, I will culture the Mycobacterium bovis in an abnormal conditions, that is like grown in high concentrations of bile for several generations. So after that, it cannot grow in normal conditions when I introduce it into a human being, because in us the bile concentration will be at a moderate concentration, but I have grown the responsible microorganism at high concentrations of bile, so it cannot grow in a normal culture conditions, but its structural epitopes are intact. So that is how they prepared Bacillus calmette-guerin vaccine or BCG vaccine. So one way to make an attenuated vaccine is to grow the microorganism in altered conditions, so that when it comes into the normal conditions, it cannot grow. So it has lost its pathogenicity, but its epitopes are intact. So another way of producing the attenuated vaccine is by the example of oral polio vaccine, which is also called as Sabin vaccine. So this particular Sabin vaccine contains 3 strains of polio virus, all of which were grown in monkey kidney epithelial cells. So this is one way to make the organism attenuated, so that when it is introduced into the human being, it cannot grow, or it will lose its pathogenicity, but its structural epitopes are intact. And also remember, oral polio vaccine, as all of you know, will be administered at least 3 times, because it contains 3 strains of polio virus. When I administer for the first time, you will be getting immunity against one of them. In the subsequent administrations, you will get second and third strains of it. So at the end of the 3 immunizations, you will be ready with immunity of all the 3 strains of polio virus. But remember, here again, that is the problem associated with the oral polio vaccine is it contains a live but weakened microorganism, that is the polio virus. So it may revert back. That is the reason why the developed countries are not using this vaccine. But the developing countries are using this vaccine because of its effectivity. So they need to eradicate the polio, so they are going with it because it is producing highly effective results. Right? So here we have discussed about 2 things. One is Mycobacterium bovis, responsible for TB, and the vaccine is BCG. The other is oral polio vaccine or Sabin vaccine, because of its developer Sabin. Another type in whole organism vaccine is inactivated vaccines. Till now we have seen about the attenuated version of vaccines where the microorganism is weakened, whereas here in inactivated vaccines or killed vaccines contain the microorganism which is permanently killed by treating it either with heat or with chemicals like formaldehyde. So the formaldehyde kills the microorganism, leaving the structural epitopes intact. Remember, whatever it is, the structural epitope should be intact so that it can produce an immune response when it is injected into the individual. So the example of this kind of a vaccine is Salk vaccine, which is an inactivated polio vaccine. We have already seen oral polio vaccine, which is developed by Sabin, and this is another vaccine developed by Salk, which contains an inactivated version of polio virus. But again, the problem associated with it is one of the batch of the vials got reverted back. So rather than alleviating from the polio, it has produced polio in one batch of US people. Whole organism vaccines, whether they are attenuated or inactivated, they are producing a problem. Attenuated vaccines have a problem of reversion, whereas an inactivated vaccines are also reverted in one case. So scientists have devised methods rather than using whole microorganisms. They are now more concentrating on the epitopes that are possibly responsible for the disease. So in that category, we have the first and foremost vaccines are purified macromolecules that could be act as vaccines. The best example for that is tetanus toxoid, and I will tell you more about them in the subsequent classes. So here, do remember, whole organism vaccines are creating a problem, because of which we are shifting our focus onto the purified macromolecules or epitopes that could be used as vaccines. And in the same way, because of the developments in the molecular biology and recombinant DNA technology, they have devised another whole set of new vaccines that are DNA vaccines, multivalent subunit vaccines, and recombinant vector vaccines, which are all focused only to the particular epitopes but not the whole organism. You will come to know about it in the subsequent classes. So here in this first video lecture, I would like to highlight up to this point. Do remember, the whole organism vaccines are posing some threat, because of which the focus has been shifted to epitopes. Alright. So we will, in the next class, we will discuss about these. We will discuss about the advances in the vaccine development by taking examples of purified macromolecules, DNA vaccines, and multivalent subunit vaccines, etc. Thank you all for your patient listening.